Okay, so as a philosopher, I'm sure you're always thinking about, how we know what we know. But have you ever considered like how these really deep questions about knowledge actually pop up, even in something like a biology lab? Welcome to the deep dive.
Where we do exactly that.
Yeah, today's going to be a really interesting one. We're diving into the philosophy of, get this, biological experimentation.
That's right. And for this one, we're going to be looking at a super fascinating article from the Stanford Encyclopedia of Philosophy.
That's right, the Stanford Encyclopedia of Philosophy.
Yeah, and, a lot of times in the philosophy of science, we're thinking about, like, the big theories.
Yeah.
Relativity, evolution, that sort of thing. But today, we're really zeroing in on experiments.
Yeah, experiments.
Because, in biology, experiments are really the key to building knowledge.
Absolutely. And I think for you, our listener, especially if you come from a philosophy background, this is going to be a really rich deep dive.
Yeah. We're going to be wrestling with some really fundamental concepts. I mean, like causality, evidence, the whole idea of rationality in science.
Rationality, right. And in the end, what does it even mean to say we know something in biology?
Exactly. And the really cool thing is that we're going to be seeing how, these kind of abstract philosophical ideas actually play out, like in the nitty-gritty world of a biology lab.
Right, in the everyday work of scientists. So in our source material, we're going to be looking at a whole bunch of stuff, how biologists figure out cause and effect, including these things called Mills methods.
Right, Mills methods.
And then we'll tackle those really tricky questions around these so-called crucial experiments and something called Duham's predicament.
Oh, yeah, Duham's predicament, classic.
It is a classic. And then we'll get into model organisms and those really complex experimental systems.
And how these things kind of end up shaping the questions that biologists even ask.
Exactly. Like the tools you use change the kind of science you can do.
It's wild.
It is. And then we'll wrap things up by looking at how scientific ideas in biology change over time.
And that constant question, like, can we really trust the data?
Right. So our mission today is to really dig deep, to figure out how biological knowledge is actually built, through these experiments. We're going beyond like the simplified textbook explanations and getting into like the really philosophically interesting details.
Yeah, the messy, the complicated, the exciting.
Exactly. So let's jump right in. Let's start with the basics. How do experiments in biology actually help scientists figure out what causes what? Because it's got to be more than just testing theories, right?
It totally is. Like a lot of times experiments in biology, they're how you find those causal links in the first place. And our source talks about this idea called Mills' method of difference. I think as a philosopher, you'll love this one. It's got that nice, clean logic to it.
Right.
And it really fits in perfectly with experimental biology, where we can actually set up these exact conditions that he's talking about.
Oh, yeah. I remember, Mill. It was something like, if you have two situations and they're the same in every way except for one thing.
And the phenomenon happens in one and not the other.
Right. Then that one difference, that's got to be the cause.
Boom. Nailed it.
It almost feels like those logic puzzles, those syllogisms.
It really does. And our source even gives this great example with, like, testing antibiotics. Yeah. So you've got these two basically identical bacteria cultures.
Right. Keep it simple.
Yeah. And you add the antibiotic to one. That's the treatment group.
Right. Treatment group.
The other one just gets the buffer solution. No antibiotic. That's your control.
Right. So the only difference between the two is that one's got the antibiotic, the other doesn't.
Exactly. And everything else is the same, right?
Yeah.
Like temperature, nutrients, all that.
Okay, controlled.
Totally controlled. So if you see that the bacteria only stop growing in the dish with the antibiotic.
Well, then the antibiotic has got to be causing that growth inhibition.
Exactly. That single difference, it isolates the cause.
It's like that one variable popping out.
Popping out, yeah.
And, thinking back to Mill, he saw this as inductive reasoning, right? Like You're going from these specific cases to a general principle. But the source points out that you could actually frame it deductively.
Right.
Which I think is really interesting from a philosophy of science perspective. Like, how does that deductive version work?
Okay, so we've got to start with some, fundamental assumptions.
Right. Lay the groundwork.
Yeah. So first off, let's say those bacteria cultures, they really are identical at the beginning.
Like no hidden differences.
Exactly. They're homogeneous, as the scientists would say.
Right.
And then we see what happens. Antibiotic, no growth. No antibiotic growth. And we're also assuming this kind of deterministic cause and effect relationship.
Meaning, if there's an effect, there's a specific cause.
Right, right. No spooky action at a distance here. So if we see the effect in one situation but not the other, and they started the same, the cause has to be something that's only in that first situation.
The one with the antibiotic.
Exactly.
Okay, that's making sense.
And if we say, okay, there are no other sneaky variables messing things up, no confounders.
Like little, like, hidden factors.
Right, lurking in the shadows. And the effect, like the lack of growth, isn't causing itself, which is, kind of a weird thought, but we got to rule it out.
Right.
Then logically, the antibiotic is the culprit, or at least part of the culprit.
Okay, so by adding in these assumptions, like the cultures are the same, there's this clear cause and effect relationship, no confounders, and the effect isn't like self-causing.
We can actually turn that cause and effect thing into a deduction.
Right, like a pure logic problem.
Yeah, but like the source points out, we haven't gotten rid of uncertainty.
Right, we've just shifted it
Exactly.
to the assumptions.
Exactly. So now the question becomes, like, how good are those assumptions? Are the cultures really identical? Are we sure there's nothing else affecting the bacteria?
Right, that's the really deep philosophical question, isn't it?
Totally. It's like, how can we really justify these assumptions?
Yeah.
And the source kind of says, there's no rock solid, like, philosophical proof for them.
Right.
But they work. They're baked into the methods that biologists use. And like those methods as a whole have been super successful. So it's almost like this pragmatic justification.
Right. Like if it works, it's probably true.
Something like that.
Yeah.
Which is a really interesting idea, philosophically speaking.
Yeah, it is. And it's not just historical stuff, right? Like
Right.
Mill's methods aren't just sitting in some dusty philosophy book.
Oh, totally not. I mean, they've been like refined, expanded, turned into these super sophisticated frameworks for figuring out cause and effect.
Right. Upgraded.
Yeah, and they're not just for theoretical stuff. Like scientists have actually used them to like analyze and understand some major scientific breakthroughs.
Oh, really?
Yeah, yeah, like the discovery of the urea cycle.
Oh, wow. Yeah, that's fundamental stuff.
Totally fundamental. And philosophers have like taken these generalized methods
Yeah.
and, kind of dissected the experiments that led to that discovery.
Right. It's almost like reverse engineering the science.
Exactly. Figuring out the logic behind the breakthroughs.
So cool. But, like the source says, these methods don't just magically create knowledge.
Yeah, you can't just like throw observations at them and expect them to spit out the answers.
Right. There's got to be something to work with.
Exactly. You need some kind of initial idea about cause and effect to even get started.
Yeah.
Like in that antibiotic example, you have to assume that the cultures are basically the same to begin with.
Right. Or else the whole thing falls apart.
Yeah.
Okay. So we've been talking about like figuring out if X causes Y, but then the source introduces this whole other idea.
Oh, yeah. Mechanistic constitution.
Yeah.
And those like interlevel experiments.
Right. So it feels like we're going beyond just simple cause and effect.
We are. Like in the old days, philosophers of science were mostly focused on those direct causal links between things. But a lot of biology today, it's about understanding mechanisms.
Mechanisms.
Yeah, like the actual like inner workings that make something happen. It's not just knowing that A causes B, but understanding the whole chain of events in between.
So instead of just observing like when calcium flows into a neuron, it releases neurotransmitters.
Right.
We want to know how those calcium ions are actually doing that. Like what proteins are they interacting with?
Totally. What steps are happening? It's like instead of just looking at the inputs and outputs of a black box, we're trying to open it up and see all the gears turning.
So it's about figuring out how the system is actually put together. And that's where these interlevel experiments come in.
Right.
How do biologists actually use experiments to figure out how those parts like contribute to the whole?
So there are two main ways. You can go bottom up.
Bottom up.
Yeah. So you mess with something at a lower level and see what happens at the higher level.
Yeah.
Like in the neuron, you could block the calcium from flowing in.
Okay. And if that stops the neurotransmitters from being released?
Then you know that calcium influx is a necessary part of that mechanism.
Right. Right. Makes sense. And then there's the opposite, right?
Yeah, you can go top down.
Top down. So you change something at the higher level and then see what's going on with the parts.
Exactly. Like you could stimulate the neuron to release more neurotransmitters.
And then check if the calcium influx changes.
Right on.
So it's this idea of like manipulating things both ways. And if you can reliably change one level by tweaking the other, that's a pretty good sign that they're part of the same mechanism.
Right. It's like this mutual manipulability thing.
Yeah.
It's a big deal for a guy named Craver who, like, argues that this back and forth is key for figuring out how things are constituted. But the source also says there's some debate around this,
Right,
right? Like,
right.
philosophically, there are some challenges.
Oh, of course it's philosophy.
Right. So one worry is that when you mess with things, especially at different levels, you're probably affecting more than one variable.
Right. So it's hard to isolate the exact relationship you're looking for.
Exactly. It's like you're using a hammer when you need tweezers.
Uh-huh.
So some philosophers think we might need something more than just Mill's methods.
Like what?
Well, maybe something like abductive reasoning.
Right, where you kind of propose the best explanation for what you're seeing.
Right. So figuring out these mechanisms, it's trickier than just simple cause and effect.
Right. Okay, so we've got a good sense of how biologists investigate those cause and effect relationships and how they dig into the mechanisms of those relationships. But let's turn to this really fascinating concept of crucial experimental evidence. You know, those textbook examples where one experiment supposedly settles a major debate. But is it really that simple?
That's what Duham's predicament comes into. Like, throw a wrench in the works.
Right, Duham's predicament.
So he was this philosopher of science, and he basically argued that truly crucial experiments, they're impossible.
Impossible.
At least in physics. Because even if an experiment seems to, like, totally rule out every other explanation you can think of,
Right.
you can never be sure that there isn't some other explanation out there, some crazy new idea that no one's even thought of yet.
Right. So we can never be 100% sure we've got it all figured out.
Exactly. It's kind of humbling,
Okay.
like no matter how much we think we know, there could always be something more. And our source uses this debate about oxidative phosphorylation as a really good example of this.
Oh, yeah. The whole, like, how cells make ATP thing.
Yeah, ATP, the energy currency of life.
Right. So can you walk us through that one?
Sure. So one of the big ideas was that there was this high energy molecule, this chemical intermediate that was like directly linking electron transport to ATP synthesis.
Okay. Kind of like a middleman.
Exactly. But like no matter how hard they looked, no one could find this molecule. And then there was Peter Mitchell.
Mitchell. Right.
He had this radical idea, this chemiosmotic mechanism. He said, look, it's all about this gradient of protons across the mitochondrial membrane. That provides the energy for ATP synthesis, no need for a middleman molecule.
Okay. Bold idea.
Super bold. And at first, Mitchell had some evidence. He showed that mitochondria could pump protons across their membranes. But a lot of scientists weren't convinced.
Right, because they could just say that the proton pumping was just a side effect, right? Like the real energy transfer was still happening through this mystery molecule.
Exactly. It's like trying to solve a puzzle with missing pieces. Both sides could point to data that kind of fit their view, but nothing was, like, definitive.
Uh-huh.
And then came this super clever experiment by Racker and Stokenius.
Racker and Stokenius, they're the heroes of the story.
Totally. So they basically built these artificial membrane sacs, like super simplified systems.
Okay, like a model of a mitochondrion.
Right. And they only put two things in there. ATPase, which is the enzyme that actually makes ATP, and this protein called bacteriorhodopsin, which pumps protons when you shine light on it.
Okay.
So they shine the light, the proton pump goes to work, creates a proton gradient, and guess what?
ATP is made.
ATP is made. But here's the thing. There are no respiratory enzymes, no mysterious chemical intermediate, just the ATPase and the proton gradient. It's like the most minimal system you can imagine.
Right. So it seems like this experiment is saying, hey, you don't need that mystery molecule. You just need the proton gradient, and the ATPase can make ATP.
Exactly. And a lot of people see this as the crucial experiment that, like, proved Mitchell's theory. And, like, our source even talks about it in terms of, like, eliminative induction. You've got these two possible causal models, and this experiment rules out the one with the chemical intermediate.
So case closed.
Well, not quite.
Yeah.
Because Duham's still lurking in the background.
Right. That pesky Duham.
He'd say, sure, this experiment shows that a proton gradient can make ATP in this, like, simplified system. But what if there's some other mechanism happening in a real mitochondrion? Some crazy, complicated thing that we haven't even thought of.
Right, like there's always the chance we're missing something.
Exactly. So our source kind of argues that the real power of the Racker-Stokenius experiment wasn't about absolute proof.
Okay, so what was it about?
It was about the method. Like, this experiment was super well controlled.
Right.
Everything was defined, purified. It was really hard for the chemical intermediate folks to say, oh, there's some other thing going on here that's making the ATP. So it wasn't a knockout punch, but it shifted the burden of proof.
Okay, it raised the stakes.
Exactly. It made the chemical intermediate idea way less likely.
Interesting. And the source also talks about that super famous Messelson-Stahl experiment, the one about DNA replication.
Oh yeah, another classic.
What can that one tell us about crucial experiments in biology?
So remember, there were these three ideas about how DNA replicates.
Right, conservative, semi-conservative, and dispersive.
Right. And Messelson and Stahl, they wanted to figure out which one was right. So they did this really clever experiment where they used heavy nitrogen to label the DNA, and then they watched what happened to the density of the DNA after it replicated.
Okay.
And the results were like super clear. After one round of replication, they saw DNA with an intermediate density, which ruled out the conservative model.
Right, because that model would have predicted two separate bands.
Exactly. And then after two rounds, they saw two bands, one intermediate and one light, just like the semi-conservative model predicted.
So semi-conservative model wins. Case closed.
Well, again, it's not that simple. Even Messelson and Stahl, they were careful. They said, look, our results only show that the nitrogen atoms are distributed pretty evenly between the two strands. They couldn't rule out, like, some weird modified conservative model where heavy and light DNA were somehow stuck together end to end.
Right, because that would still give you that intermediate density.
Exactly. And there's another interesting twist. Turns out, the way they were handling the DNA, they were actually breaking it up a bit. They didn't realize it at the time.
Oh, interesting.
So even though the results looked really clean, there were these hidden factors at play.
So once again, even this textbook crucial experiment wasn't totally perfect?
Nope. Our source actually suggests thinking about it in terms of inference to the best explanation, or IBE.
IBE.
Yeah. The semi-conservative model, it just explained the results better without needing any extra assumptions.
Yeah.
It was the most elegant, the most parsimonious.
Right. So these landmark experiments, they're super important.
Totally.
But the idea that one experiment can definitively prove something and eliminate all the other possibilities, that's probably a bit of a fantasy.
Yeah, I think Doolin was right to be skeptical. Science is messy. There are always things we don't know.
Right. So these experiments, they give us strong evidence. They make some explanations more likely than others, but they're not these knockout punches that end the debate.
Science is a process.
Right. And sometimes it's more of a points decision.
Exactly.
Okay, so it's clear that experiments do a lot more than just test theories.
Right.
The source makes this really cool point about how experimentation has a life of its own. And they highlight this whole idea of model organisms and those complex experimental systems. It seems like we're moving away from a theory-driven view of science and towards something where the experiments themselves are driving things.
Totally. Like traditional philosophy of science, it's always been about how theories get tested. But historians and philosophers of biology, they're saying, hey, experiments are doing a lot more than that.
Right.
Like a lot of the work that goes on in a lab, it's not about confirming or denying some grand theory. It's about exploring, finding new things, maybe even figuring out what questions to ask in the first place.
Right. So the experiments themselves are helping to shape the research.
Exactly. And model organisms are a huge part of this.
Right. The fruit flies, the worms, the bacteria.
Yeah. They're like the workhorses of biology. But it's kind of weird, right? Like we're trying to understand all of life by studying this tiny little sliver of it.
It's a paradox.
It is, and it's a big one for philosophers who are thinking about the unity of science.
Right. Can we really generalize from these very specific organisms to the whole of biology?
Exactly. And the funny thing is, the choice of which organism to study, it's not always based on some grand theoretical idea. Sometimes it's just about practicality.
Right.
Like, is it easy to breed? Does it have a short generation time?
Can you keep it in a lab without too much trouble?
Exactly. There's this great study by Kohler where he looked at the history of Drosophila genetics, and he called the fruit fly system a breeder reactor. It's like this machine for churning out new genetic mutants. And the focus wasn't always on testing some specific theory. It was on making all this experimental material and developing the tools to analyze it.
So the model organism itself becomes a kind of research tool.
Totally. It's a way to generate new puzzles, new possibilities.
Yeah.
Even if we don't have a perfect theory to guide us yet.
Right. And there's this element of chance in which organisms become models, right? It's not always the most theoretically perfect species.
Oh, yeah. Sometimes it's just historical accident. Like someone was working on a particular organism for some other reason, and then bam, it becomes the model for a whole field. But then this raises the question of okay, how do we know what we learn from these models applies to other things? Like, can we really extrapolate from fruit flies to humans?
Right, that's the big question. And the source talks about this idea of the extrapolator's circle. It's this like chicken and egg problem.
Right, like to be sure we can extrapolate, we need to know that the model and the target are similar in the ways that matter. But to know if they're similar, we need to know about the thing we're studying in both of them.
Right, it's circular.
Totally. So, like for something like the genetic code, we could use evolutionary arguments to say, okay, this is probably universal. But for other things, it's a lot trickier.
And the source mentions steel, right?
Yeah, Steele talks about this thing called comparative process tracing. It's about carefully comparing the steps and components of the mechanism in the model and the target, to see where they're similar and where they're different.
Okay, so it's about getting really granular, really specific.
Exactly. And then there's the whole thing with gene sequences. Like when you see similar genes in different organisms, that's a good sign that they might have similar mechanisms because they both inherited it from a common ancestor. Like with those homeobox genes, they're super important for development, and you find them in everything from flies to people.
Right, so that shared genetic blueprint is evidence that the underlying mechanisms are probably similar.
Exactly. And whether these kinds of extrapolations are like rock solid or more like, educated guesses, that's still up for debate. But the point is, these model organisms, they're not just passive things we experiment on. They're actually shaping the whole field of biology.
It's a really interesting idea.
It is. And the source even talks about like whether model organisms can be seen as a kind of theoretical model themselves.
Whoa.
Yeah, like are they simplified representations of like the broader biological world?
That's really making me think.
Right.
Okay, so we've talked about model organisms, but the source also brings up this idea of experimental systems. It seems like it's related to model organisms, but a bit different.
Yeah, so this is where Reinberger comes in. He's like super into experimental systems. And he sees them as these really specific setups, with particular materials, protocols, instruments, everything that's needed to investigate a specific question.
Okay, so it's like the whole ecosystem of the experiment.
Right, the whole shebang. And Reinberger's got this great quote. He calls them systems of manipulation designed to give unknown answers.
Wow.
I know, right?
It's kind of poetic.
It is. And he says these systems, they're like the fundamental units of research. Like you build the system first, and then the knowledge comes out of it.
So it's not always about starting with a theory and then designing the experiment.
Right. Sometimes it's the other way around. And the source talks about that in vitro protein synthesis system.
Right. The one that, helped crack the genetic code.
Exactly. But the funny thing is, that system, it wasn't originally meant for that. It was supposed to be for studying cancer cells.
Oh, interesting.
So it's like the system led them to this unexpected discovery.
Right. It opened up a whole new area of research.
Totally. And Reinberger talks about epistemic things.
Yeah.
These are the things that are like the focus of research within the system. But at first, we don't really know what they are.
Okay.
Like the ribosome in the early days. They knew it was important, but they didn't really know what it did.
Right. It was a mystery.
Totally. But as you refine the system, these epistemic things, they become more like technical objects. We understand them better, we can manipulate them, we can use them to learn even more.
So the system is driving the knowledge, but it's also evolving as our knowledge grows.
Yeah, it's a cool back and forth. But then philosophically, there's this question of, what are these systems really?
Right. Are they just like physical things, like the equipment and the chemicals?
Or do they have a conceptual side too? Like the ideas and theories that guide how the system is built and used.
Right, because a bunch of lab equipment by itself, that's not an experimental system.
No, exactly. It's the scientist's mind, their ideas, their goals that bring it all together.
That's deep. Okay, so we've covered a lot about how experiments are done, the different things they can achieve. But now let's talk about how scientists, as a community, decide which theories to keep and which ones to toss. Which brings us to the question of, rationality in science.
Right. This is one of those classic philosophy of science questions.
Yeah, is science really this perfectly rational process where evidence always wins the day? Or is it messier than that?
Right. And the traditional view, the Popperian view, where it's all about falsification, that doesn't quite fit with biology, especially with all this stuff we've been talking about, where experiments aren't always about testing some grand theory. So how do scientists actually choose between different ideas, especially when the evidence is complicated or maybe even contradictory?
Right. And the source talks about the oxidative phosphorylation debate again, right?
Yeah, they point out that even in that one case, there are all these different interpretations of how the debate was settled. It's like you can tell the same story in different ways depending on your philosophical perspective.
So it's tough to prove that science is purely rational, but it's also hard to say it's all just social and political.
Right. And this is where social epistemology comes in.
Social epistemology.
Yeah, it's about how knowledge is made within communities. It's like acknowledging that science is a human activity with all its messiness and social dynamics.
Right.
But at the same time, it can still be rational.
Okay, I like that.
Like the community can work together, debate, share ideas, and in the end, come up with reliable knowledge.
Even if individual scientists aren't always perfectly rational.
Exactly. And the source uses the history of genetics as an example.
Oh, yeah. Early 20th century genetics, there were all those different schools of thought.
Right. But the one that really took off, the one that kind of won the day, was the Morgan School, the Drosophila folks.
Right.
And the argument isn't just that their theory was the best.
Okay, so what was it?
It was that their whole approach, their theory, their experiments, their model organism, it was all just super successful.
Like, it worked.
It worked. It produced results. It opened up new areas of research. It connected with other fields like evolutionary biology. So it wasn't just about one theory beating another. It was about this whole way of doing genetics.
Right, like the research program itself was selected for.
Exactly. And that connects to like Cood's ideas about paradigms and this like emergent social rationality. Yeah. It's the community, the collective practices that drive scientific progress.
Even if no single scientist is making like perfectly rational decisions all the time.
Right. It's like this like wisdom of the crowd thing.
I like that.
Yeah.
Okay, so we've talked about how experiments work, how they shape research, how scientists make decisions.
Right.
But even the most carefully done